Hello and welcome to the 14th in my series of presentations on the conflict between science and creationism. Today I'm going to be talking about the subject of genetics, the subject that investigates the chemical code in our body's cells that encodes all the information used to describe living things. In particular, I'll be looking at a number of claims made in the presentations of the infamous young Earth creationist Kent Hovind. This means that I won't be covering the whole topic, and I won't even be covering it in any sensible order. I'll just cover the topics that Hovind mentions. Genetics is another great subject to focus on because it is absolutely full of cast-iron proofs that evolution is real. The best bit is that the creationists have nothing here whatsoever. Absolutely everything is completely and undeniably against them, so they will never bring up genetics in general. Your job is to keep the discussion on genetics for as long as possible, because it should provide the basis for any comprehensive disproof of creationist claims. Not just young Earth creationism, but everything up to and including intelligent design completely crumbles in the light of genetic evidence. It's fairly obvious, I think, that genetics is a complicated subject, and although they very rarely attempt to resort to genetic arguments, it's easy for a well-read creationist to come up with some pretty powerful counter-arguments that, although they're not actually valid, will silence all but the most learned opponents. One of the great problems with debating at this level is simply that the creationists have some very sophisticated arguments indeed, akin to the man who attempts to justify the existence of Father Christmas by invoking relativistic time dilation and parallel universes. Extremely stupid or downright false ideas can sound very convincing when wrapped up in advanced, sciencey sounding terminology. Of course, you should never resort to this tactic yourself because A, it's dishonest and will just drag you down to the level of the creationists, and B, it will backfire badly if they point out the trick that you've just played. Honesty is your main weapon. If you can be totally honest yourself and point out all the times when your opponents lie and deceive, then you will gain a hefty advantage. So let's get started. DNA is the chemical that encodes the famous language of life that exists within the cells of every living creature. Its familiar patterns of the four base chemicals, adenine, cytosine, guanine and thymine, contain the complete set of inherited information for how to create a new version of you. Not all of you, of course. DNA doesn't encode your experiences and specific personal memories and developmental quirks, but it's a pretty thorough description of you at a biological level. However, the part of your genome that codes for proteins, the major function of DNA, is actually just a tiny minority, less than 2% of your entire genetic code. The remainder used to be known as junk DNA, but nowadays is known more formally and more correctly as non-coding DNA by experts in the field. Non-coding DNA may not code for the building blocks of certain proteins, but that doesn't mean that it's entirely useless. Creationists attempt to make up a straw man for the scientific argument here by claiming that scientists believe that all non-coding DNA is totally useless. Then they quote a study, by a scientist of course, which shows that this isn't true. Doesn't matter which study, there's loads of them, but whatever they choose, the argument is that the so-called junk DNA isn't so useless after all. Science is wrong and therefore the Earth is 6,000 years old. I'm sure you follow their logic. The most major flaw, of course, in that logic is that they're claiming that scientists believe that all junk DNA is useless, and then they quote a scientist who says it isn't. But that's probably the least of their problems. In reality, the picture is much more subtle than this. Indeed, the majority of DNA doesn't appear to do much at all, but that doesn't mean that it's useless, of course, though it may not code for proteins. A 2007 study showed that as much as 80% of the non-coding DNA may well be transcribed in the cell, but that doesn't imply that it actually has a purpose, because the results may simply be thrown away. What can't be doubted, however, is that there is a lot of wasted information in our genetic code. There are large sequences of the same identical patterns repeated many, many times in succession. 20% of the genome is never even transcribed, and hence never read by anything. The genome is hardly the ultra-efficient, concise and accurate blueprint that you would expect were it to have been designed. Of course, evolution explains this perfectly, which is all the more impressive because Darwin and Wallace knew nothing about genetics when they wrote down their theories. Ultimately, DNA copying is a blind and short-sighted process. The mechanisms by which this work can be, and are, hijacked by viruses to do their work, and those viruses often get inserted into our DNA. We'll talk about that later. In addition, this blind process makes mistakes, and those mistakes accumulate over time because there isn't a strong selection pressure towards a short, concise genome. Those mistakes vary from species to species, but there are some remarkable similarities, where the exact same lengthy mistake can be seen in genomes of related species, but not in others, and the degree to which those areas of code differ is directly related to how recently we believe those organisms shared a common ancestor. That's basically impossible for a creationist to explain without evoking the mischievous designer hypothesis, where the designer, in this case God, deliberately inserted specific features into the genomes of animals in order to make them look like they evolved. But let's look at those features in turn. An endogenous retrovirus, or ERV, is an ancient virus that once infected our distant evolutionary ancestors and wrote itself into our genome. As I mentioned on the previous slide, it does this by hijacking our own molecular apparatus in order to serve its own purpose. The retroviruses use reverse transcriptase in order to convert their own RNA into the DNA of our own molecular code, and then insert themselves into the genome as it's replicated in cell division. Though this sounds like science fiction, these sections of genome are very well studied and can be compared between related species. We share several of these with our most recent evolutionary ancestors, and there are several thousand in the human genome in total. Unsurprisingly, the more distant the evolutionary ancestor, the fewer endogenous retrovirus segments in the genome that we have in common. In addition to this, ERVs give us a nice molecular clock. Individual endogenous retrovirus sequences are mutated over time, just like the rest of our DNA. As these came from a single ancestor, we know that for every pair of creatures who have a similar ERV sequence, there was once an ancestor species that contained an earlier version of this segment of DNA. The alternative possibility being that two species both gained an almost identical ERV in an almost identical place in their genomes entirely separately. The chances against this are obviously astronomical. So given that we know that a particular section of DNA in two species came from a common ancestor, we can count the differences and, using known frequencies of genetic mutations, we can estimate the approximate time at which these two lineages diverged. The more differences, the longer since the common ancestor. And this gives yet another set of stunning proofs for the evolutionary theory. Hemoglobin is a protein that makes up almost all of the dry mass of the red blood cells in our blood, which transport oxygen from our lungs to every other part of our bodies. Hemoglobin consists mostly of proteins, which are folded chains of amino acids, also called polypeptides. The precise sequences which make up these polypeptides are determined by the genetic code of the creature which you're investigating. The genes for hemoglobin very precisely describe how it's constructed, and therefore its properties and function. The genes which code for hemoglobin usually differ between species, and as you would expect by now, the number of differences increases with the evolutionary distance between these species. For example, the most common hemoglobin sequences in humans and chimpanzees are nearly identical, as you would expect. One way to analyse these differences is to look at the individual alpha and beta chains of hemoglobin, and the various locations that code for a specific amino acid. Let's take glutamic acid, as it's been well studied because of the role of glutamic acid in hemoglobin and the disease sickle cell anemia. Each of these two chains, the alpha chain and the beta chain, which we can think of as subunits of the hemoglobin molecule, is very well understood. Alpha chains consist of 141 amino acids and beta chains consist of 146 amino acids. Glutamic acid can be coded either by the codon GAA in the genetic code, that is guanine, adenine, adenine, or GAG, guanine, adenine, guanine. There are 12 locations in the alpha and beta chains which make up most normal hemoglobin in adults, where glutamic acid is found. In each case, we could see either GAG or GAA coding for glutamic acid, as it's functionally identical. However, what we notice is that in humans, chimpanzees and monkeys, it's always the same variant in each location. The chances against any one random pair of species using the same variant in every location is 1 in 4096, but the chance of each pair of species across such a wide range sharing the same pairs is essentially zero. Of course, this sounds like a creationist argument, but it's exactly the opposite. The chance of randomly creating a species with the same arbitrary and pointless description of a specific molecule is tiny, but it's exactly what we would expect from an evolutionary model. Of course, the creationists just retreat to one of their two favourite answers here. Either God made it to look like it evolved, but it didn't. Actually, that's a very rare argument that they hardly ever use. Or, much more likely, God had a common toolbox which he used across a wide range of creatures. Hence the similarity. Very well. So in that case, why did he not continue to use exactly that same toolbox across all creatures? Why is it that the glutamic acid codon starts subtly varying once we look at more distantly related creatures? And yet again, they vary with the distance of the relationship, or how recently these creatures shared a common ancestor. There is yet again only one sensible explanation of this fact, and it's evolution. We've already met hemoglobin on the last slide, so here's another interesting fact about the hemoglobin alpha chain that I mentioned before. If you remember, I said that the alpha chain contains 141 amino acids in a particular sequence. It is distortions of this sequence in humans that cause conditions such as sickle cell anemia. Now, there are 20 different amino acids to choose from, which are coded by three-letter genetic sequences called codons. So for 141 amino acids, there's 20 to the power of 141 different combinations, which is an astronomically enormous number. Creationists claim that this is a massive disproof of evolution because the chances of all these being randomly selected in exactly this order is essentially zero. However, they're missing the point, as ever, because of the number one mistake that creationists make. They assume that evolution is random, but it isn't. For a start, some strings are more likely to evolve than others because certain combinations of amino acids form together more readily than others. Secondly, the current form is not the only plausible form. Sure, there are deficiencies if you swap round certain base pairs, but it's not at all clear that the other combinations wouldn't also work equally well, or perhaps better, than the 141 amino acids that we use in the human genome. In fact, biochemists have done this calculation, and there are an estimated 20 to the power of 126 usable forms of the alpha chain amino acid sequence, and that's just assuming that it has to be 141 amino acids long. So the probability of forming any useful chain is only 1 in 20 to the 25, which is still huge, but if you consider evolutionary timescales and the amount of molecules involved, it's actually not that unlikely. When you further add in the fact that certain chains form much more rapidly and the fact that smaller base units of the existing chain exist which actually do perform useful functions, so therefore act as stepping stones for the alpha chain as we know it, then this rapidly becomes much more likely. In fact, the similarity of hemoglobin strings in humans and other primates is a very strong evidence for evolution. It shows the fact that we've all evolved from a common ancestor, rather than all appearing at random in separate creation events. Again, creationists use the God's common toolbox argument, but this falls down for exactly the same reason as before. Why did God use a very similar toolbox for humans and chimpanzees, a slightly less similar one for slightly less related animals, and so on? That makes no sense at all. Surely either there is one optimum molecule for the transport of oxygen throughout the bodies of animals, in which case we should all have exactly the same sequence, or there are many optimal sequences, in which case it makes no sense to see that this smooth relation exists between the evolutionary distance and the hemoglobin chain. To summarise, either God should use the same chain for every creature because it's the best way of doing things, or he shouldn't care which chain to use. Making a pattern that precisely mimics what you would expect from an evolutionary process, and from nothing else, makes no sense at all under the intelligent design hypothesis. Cytochrome C is an enzyme that is found in all living creatures. It is a component of the electron transport chain in mitochondria, which is vital to the proper working of each and every one of your body's cells. The hypothesis is that all creatures inherited the pattern for manufacturing this enzyme from a common and hence very ancient ancestor. And so we would expect the cytochrome C sequences in a range of creatures to vary by an amount that is related to the amount of time since they last shared a common ancestor. And yet again, genetics comes up with the goods here, and we have another solid proof of evolution. As you can see from the table on this slide, which shows a number of differences in the cytochrome C protein from that found in humans, compared with the number of creatures. The number of differences increases as the creatures become more unrelated to Homo sapiens. Hence, there is only one difference with the sequence found in chimpanzees, but there's 13 with a dog, 18 with a chicken, 31 with a tuna fish, and 56 with yeast. So let's summarise what we've covered today in the topic of genetics, which is without a doubt the strongest and most impressive component of our scientific evidence supporting the fact of evolution. Without any other science, genetics alone can easily vindicate this theory beyond any doubt. The addition of astronomy, geology, archaeology, radiometric dating, zoology, botany, anatomy, and all the other sciences that we'll cover in this course merely serve to solidify the evidence time and again to the point where it is utterly unassailable 1,000,000 times over. Genetics comprehensively proves that our last common ancestor with all other humans was very much older than 6,000 years ago, more like 20 or 30 times older than that. It also shows how long ago we shared a common ancestor with all other species. For example, our last common ancestor with the chimpanzees was somewhere around 7 to 9 million years ago by modern estimates. This is not a number that's open to much debate. Creationists claim that not only the Earth is 1,000 times younger than this, but also that humans and chimps never shared a common ancestor. In which case they have to explain why there are so many genetic similarities between our two species. In fact, chimps and humans share a genetic similarity that is in the high 90s%, variously quoted as 98%, 97%, sometimes as low as 95%, but still extremely high, and this depends on exactly how you measure it. A creationist has to explain this fact, which they usually do by saying that an intelligent designer would use the same method to make all his creations. In which case, as we've seen, they need to explain why he designed them in such a way that they look extraordinarily accurately like they've evolved from a common ancestor. Why be deceptive? They would also have to explain why the intelligent designer thought to replicate non-coding or junk portions of the DNA and introduce slight errors in the duplications. And they would have to explain why the same designer introduced genetic diseases into the genome, amongst many, many other things. The best thing about the science of genetics is that you can see it all happening. You can see the actual DNA molecule under a scanning tunnelling electron microscope, and you can watch the process of DNA replication as it happens. It is incontrovertible that DNA is the fabric with which our genetic code is written, and the mechanism for persisting individual traits through the generations. It is utterly incontrovertible also that the genetic codes that we are gradually unravelling for our own species and those in the world around us demonstrate beyond a shadow of a doubt that we share common ancestors with all living things. One of the greatest discoveries of this, or indeed any age, and possibly the most joyous and profoundly beautiful fact that I know of. You won't find creationists talk about genetics, except defensively, because they're aware that this subject, more than any other that I'll cover in this short course, makes their silly little delusion look petty, infantile, and laughably false. During the Dover, Pennsylvania trial between intelligent design proponents and science, the intelligent design creationists offered no defence whatsoever to any of the mountains of genetic evidence presented against them. I suggest that's because they knew they couldn't. Please do look into this beautiful subject as much as you can, because with every new study has uncovered another fascinating chapter in the story of life, and I'm sure that you'll find that as immensely exciting as I do. Well, that's all for now. As ever, there's loads more information on my website at frame.net, where you can also find a transcript of this talk, and all the following ones and all the previous ones, and you can keep up to date with my blog as well as learning about some of my other work. See you next time when I'll be talking about the science of botany, the study of plants. Sometimes we forget that evolution works on plants just as it works on animals, and by studying this enormous kingdom of living things we can learn some fascinating truths about the world. Thanks for listening.